Good morning to everyone present here. I hope you're having a pleasant day so far. I am Sharanya Dumre Patil and I will be your host. It is with great privilege and pleasure that we extend a warm welcome on behalf of Amity University Mumbai to this distinguished lecture series on biology of tuberculosis. Today's lecture will be delivered by our esteemed speaker, Dr. Ved Prakash Dwivedi sir, and the topic is novel vaccination and immunotherapeutic strategies to combat tuberculosis. This series is organized by Amity Institute of Biotechnology. In the ongoing battle against tuberculosis, the quest for effective prevention and treatment strategies is unrelenting. This lecture takes us into the realm of pioneering research, where scientists are pushing boundaries to develop novel approaches that harness the power of our immune system to combat this formidable disease. Our esteemed speaker is an expert in the field and will guide us through this landscape of innovation. Their insights promise to unveil the latest breakthroughs in vaccination and immunotherapeutics strategies, shedding light on how these advancements may reshape our tactics and in the fight against tuberculosis. That said, I would like to introduce HOI of AIB, Dr. Penna Suprasanna sir. He was the former head of Nuclear Agriculture and Biotechnology Division in BARC Mumbai and former professor at Homi Bhabha National Institute, Mumbai, India. Also, he is Dean of Research and Professor and Director of Amity Center for Nuclear Biology. I will request Penna sir to share welcome address. Over to you, sir.
Very good morning to everyone. Very good morning. Tuberculosis series hosted by Amity Institute, and we have an Dr. Ved Prakash Dwivedi, who has done pioneering work in the field of vaccines being developed for as we have to experts on this research. We have seen how serious is the disease. Therapeutic interventions are required. Vaccines being developed the world in different they have been successful, not so successful. So, or encountered in this, Dr. Vediji is here with us and I am looking forward to listen to him, and I'm sure all of you are very eager to listen. Dr. Ved Prakash. Thank you very much.
Thank you so much, sir, for taking time out of your busy schedule and saying few words. That said, I would request Dr. Shashank Kamre, sir, Assistant Professor at AIB, to introduce today's speaker. Shashank, sir, over to you.
So it is with great privilege that I'm introducing Dr. Ved Prakash Dwivedi. Actually, he's my senior in JNU and a very nice person. So Dr. Ved obtained his PhD from University of Calcutta in 2014 in the field of immunology. But after that, he served as DST Inspire faculty in the ICGP New Delhi. And he also did a postdoc from University of KwaZulu-Natal in Durban, South Africa. Dr. Ved Prakash Dwivedi also obtained various awards, such as the Earlier Career Research Award from DST SERB, Nasi Young Scientist Platinum Jubilee Award in 2019. He also got selected for the Ethical Committee of ICMR, and he has been awarded with BRICS Young Scientist Award in 22, and he has also received ICMR Shakuntala Amir Chand Prize in 2020. So apart from that, Dr. Ved has been very instrumental in basically working on the role of immunomodulators in a kind of host therapy against tuberculosis. So he has been using various natural compounds as well as small molecules, and since he is a hardcore immunologist, he can also he has been also looking at the induction of the cell-mediated immune response response as well as human immune response. And as we all know that the generation of CMI basically is a very essential thing when you look for a good vaccine candidate. And he has been also looking after the generation of the memory T cells, which are specific in response to a particular antigen, and that basically forms a basis for good vaccine candidates. So over to you, Dr. Ved.
Thanks, Professor Suprasanna, and thanks, Shranya, and of course, Shashank, for your kind words and inviting me to discuss and present my work on this platform. So should I start now? Yeah. So good morning, everyone. So today I'll be talking about some novel vaccination and immunotherapeutic approaches to combat TB. As we all know that understanding the host productive immunity against TB is really very complex. It is still unclear which antigen or which immune cells provide protective immunity against this deadly disease. So every day we are increasing our understanding to unscramble the complexity of this disease and associated immune responses. So as we all know, TB is the global pandemic disease, and as per WHO, 1/4 of the global population is latently infected with Mycobacterium tuberculosis, that is the causative agent of tuberculosis, and is responsible for millions of deaths each year. It has spread worldwide, but more prevalent in Southern and Central Africa, South Asia, and Latin America. So Mycobacterium tuberculosis mainly infects through respiratory tract, where the alveolar macrophages engulf airborne bacilli. Bacteria replicates inside the macrophages. Macrophages secrete some cytokine and chemokine in response to bacteria to activate the lymphocyte. Once they get activated, they migrate to the site of infection and form granuloma. The main function of the granuloma is to prevent the spreading of bacilli from lung to other organ, as well as concentrate the immune response at the site of infection. If the immune response is stronger, then bacteria will be contained and granuloma will be reduced. However, if there is weak immunity, then bacteria will replicate, granuloma will caseate and rupture and spill the bacilli into the airway to infect others through coughing and sneezing. So depending upon the cytokinic environment, T helper cell can get differentiated into Th1, Th2, Th17 and T regulatory cell. Although there are other T helper subsets like Th9 and T follicular TFH cells, but I'm not going to discuss them because they are very less studied in case of TB. So Th1 along with Th17 cells provide resistance to TB. However, Th2 and Treg cells assist in disease. So now the question is, how do these T cells get activated? So actually what happens that when these macrophages and dendritic cell both phagocytose the bacteria, and these dendritic cell has very less phagocytotic activity as macrophages. So they do not migrate to the lymph node, rather they transfer the antigen to other uninfected DCs, and eventually they activate the naive CD4 T cells, and after getting activated, these T cells migrate from lymph node to lung with the help of L-integrin, here in this case CD62L, and participate in the elimination of the pathogen. So now TB is present on the earth since ages, but it is still considered as global pandemic. Now, the problem lies both in its prevention, where we have only one vaccine, that is 100-year-old BCG, which is very effective in case of disseminated and meningeal TB, however, has questionable efficacy against adult pulmonary TB. On the other hand, it's a treatment which consists of multiple expensive antibiotics with no emphasis whatsoever on the host. So although there are multiple T cell subsets which are known to play a crucial role in containing MTB infection, but still the exact correlates of host productive immunity against this deadly disease is not known. So my lab mainly focuses on the generation of adjunct vaccine candidate, novel vaccine candidate. We are also characterizing various conventional and non-conventional immune cells. We are also looking at the what are the role of epigenetic modulation on the host immunity. We are also working on the formulation of various adjunct therapy with the immunomodulator. So now, if we are talking about vaccine, then we should definitely talk about BCG vaccine and what are the problem associated with this vaccine. So, BCG is very effective in case of disseminated and meningeal TB, but it has questionable efficacy against adult pulmonary TB. So recently, we and others have reported that animals primarily immunized with BCG effectively develops effector memory response and fails to induce significant population of central memory T cell responses. That is the main precursor of memory cell, which is responsible for the long-term protection against TB. So this might be one of the reasons for the failure of BCG. So there are other regions which are associated with the failure of BCG in high TB endemic region. So presence of NTM, pre-exposure with NTM, non-tuberculosis mycobacteria, can interfere with the antigen processing, and thus it can affect the maturation and transport of MSC molecule on the surface of APC, and that is the main process required for the T cell activation. So in high TB endemic region, presence of helminth infection results in the release of cytokine, that is IL-25 and IL-33, which creates a conducive environment for the differentiation of T helper 2 cell by secreting IL-4, 5, and 13. That is again, that can again assist in disease progression. So pre-existing NTM antibodies can cross-react with BCG antigen, and thus it affects the BCG's replication. And as we all know, BCG is a live vaccine. It has to replicate for its continuous activity. Moreover, BCG also induces IL-10 producing T regulatory cells, that is TR1, and that again assists in disease progression by down-regulating the protective Th1 responses. Therefore, there is an urgent need to either improve BCG vaccine or to develop novel vaccination strategy which can overcome the limitation of this present available vaccine as well as therapy. So to improve the efficacy of BCG vaccine, first we need to understand why actually BCG doesn't work. So for this, we infected a group of mice with virulent strain S37RV and vaccine strain BCG. And at different time point, we took out the lung to look at the bacterial burden as well as immune responses. So interestingly, initially both strain replicated at similar manner, but at later time point, growth of BCG got gradually diminished. So it might be the reason that adaptive immune response plays an important role for the clearance of BCG. So then we looked at the granulomatic lesion in the mice, and we observed that, yeah, BCG infected mice has less granulomatic lesion. Then we looked at the immune response, and we observed that both strain induces comparable amount of IFN gamma producing Th1 cells. However, BCG was unable to induce IL-17 producing Th17 cells. So what is the reason behind this difference? So as we all know that as compared to virulent strain, S37RV, BCG lacks several, BCG has, several mutations, and these mutations are known as region of differences. RDs, RD1 to RD9. So among these RDs, RD1 is the most dominant, prominent, and important one for providing virulency to Mycobacterium tuberculosis. So we thought whether RD1 has any role in the difference of immunogenicity in S37RV and BCG. For this, we deleted we deleted RD1 from S37RV and put back RD1 into BCG, and then we infected mice with all these strains, and then again we looked at the bacterial burden and immune responses. So we observed that del RD1 deleted S37RV and BCG were behaving similar. However, S37RV and RD1 knock-in BCG were behaving similar in terms of bacterial burden as well as immune response. So now we can say that, yes, there is a role of RD1 in the difference in immunogenicity of S37RV and BCG. So now, then we looked at the TS17 cell level and as well as TS17 differentiating cytokine in the ball fluid of infected animal, and we observed that RD1 deleted S37RV and BCG were unable to induce IL-6 and TGF-beta, that is TS17 differentiating cytokine, and so as IL-17. So now RD1 plays an important role in the difference of TS17 cells. So now we ask, even if S37RV induces TS17 cell, but still it is not providing protection. Then we went back to the literature, and we came across many literature where people had shown that yes, IL-17 might have a role in secondary infection. So for this, we again performed an experiment wherein mice were infected with these strains, S37RV, BCG, RD1 deleted S37RV, and RD1 knock-in BCG. And at day 30, we started giving antibiotics to them. And then after a short period of rest, after a short period of rest, we performed CFU to check whether any viable bacteria is present in the lung of infected animals. Then we reinfected these animals with only S37RV. And then after a certain time point, we looked at the bacterial burden and immune responses. And interestingly, what we observed that the mice which were previously infected with BCG or RD1 deleted S37RV now providing better protection as compared to primary infection. However, the mice which were previously infected with S37RV and RD1 knock-in BCG now provided robust protective immunity as compared to either control. So now we can say that yes, RD1 region enhances protective immune responses. So to further confirm that IL-17 provide protection during secondary infection, this time we performed the similar experiment, but this time we injected anti-IL-17 antibodies every 72 hours during rechallenge with S37RV to animals previously infected with S37RV or RD1 knock-in BCG. And interestingly, what we observed that treatment with anti-IL-17 abrogated the enhancement in protective immune response induced by either S37RV or RD1 knock-in BCG. So now we can say that yes, TS17 cells provide protective role for better protective immune response against secondary infection. So now we know that RD1 is having a lot of many genes. So what are the genes which are responsible for the differentiation of IL-17? So then we again went back to the literature and observed that ESAT6-CFP10 is the major protein which has been extensively studied. So we thought whether ESAT6 has any role in the differentiation of IL-17 or TS17 cells. So for this, we generated dendritic cell from femur bone marrow of the mice, and then we treated with these ESAT6 and at different time point, and then we collected the supernatant of the cells, and we looked at the cytokine level, and we observed that yes, ESAT6 was able to induce IL-6 and TGF-beta in a TLR2 and MYD88 manner. Then again, we performed the similar experiment. We isolated the dendritic cells in treated with ESAT6, and this time we co-cultured this setting with CD4 T cells derived from OT2 transgenic mice. And then we looked at the level of IFN gamma and IL-17 in the cultured supernatant. And we observed that yes, ESAT6 was able to induce IL-17 in TLR2 and MYD88 dependent manner. So now we wanted to look at the mechanism. So then again, we came across many literature, and we observed that in 2007 or 8, Baltimore group first time showed that microRNA has, microRNA regulates innate immunity. So we thought whether in this setting, whether any microRNA has any role in the, negative role or positive role in the upregulation or downregulation of this IL-17 differentiating cytokine. So we observed that yes, ESAT6 negatively regulates Mir146A and allows the induction of IL-6, which is responsible for the differentiation of TS17 cells. So IL-6 and TGF-beta induced by ESAT6 in disease promote TS17 differentiation. So to summarize this part, ESAT6 protein, which is present in S37RV but not in BCG, directs TS17 cell differentiation by inducing IL-6 and TGF-beta in disease in a TLR2 and MYD manner, and by down-regulating BR146A. So now we have proved that yes, S37RV induces better protective response than BCG itself. But for obvious reason, we cannot use S37RV as a vaccine candidate. So then what we should do? Then we came up with the idea that why can't we make a MTB mimic that can enhance the immunogenicity without causing any pathogenicity. So for this, we screen many MTB peptide for their efficacy to activate T cell response as well as protective cytokine responses. And we selected some of them and combined together with TLR ligands and absorbed the TLR2 and TLR9 ligand and absorb these molecule on the surface of liposome to facilitate their entry into the lung of mice. And then we named this molecule as PTL, peptide TLR liposome. And then we asked whether these molecules are getting absorbed in the lung. So for this, we stained this liposome containing molecule with PKS67 dye and injected into the lung of mice. And then after 48 hours, we took out the lung and prepare the section and look at the intensity of the PKS67 dye. And interestingly, we observed that these liposomes were getting absorbed after 48 hours. And this molecule we recently have patented, and now it is in national phase of various African and Southeast Asian countries. Then we ask whether this PTL molecule can enhance the vaccine efficacy of BCG. So for this we immunized mice with BCG, and these BCG immunized mice were subsequently boosted with these PTLs. And following a rest of period, we infected these mice with S37RB through low dose aerosol infection model. And then at different time point, we look at the bacterial burden as well as immune responses. So interestingly, what we observed that PTL co-immunization significantly enhanced the BCG-induced antitubercular immunity, as evidenced by reduction in the bacterial burden by more than 2 log in the lung and spleen of the mice. Again, PTL co-immunization significantly enhances the polyfunctional cytokine responses as well as memory responses, which further led to the reduction in the rate of TB reactivation, which is the major challenge being faced due to current antituberculosis therapy. So this study was just published. This study was published in JCI Insight. Yes, we perform other experiment to validate whether this PTL molecule can enhance the efficacy in different setting, like can it reduce the TB reactivation, reinfection. So we performed every experiment. Now we performed some experiments in human. So now fortunately we got lucky, and now this molecule is going in different phases. So hopefully we'll get some data in near future we can discuss. So moving on to the second part, where we are involved in the formulation of adjunct therapy. So as we all know, the present available antituberculosis therapy consists of multiple expensive antibiotics. So frontline anti-TB drug is isoniazid, rifampicin, pyrazinamide, and ethambutol. It is very lengthy, and due to length of treatment, once a patient starts feeling better, they withdraw from the therapy, and that is the main cause of emergence of drug-resistant variants. It also induces various side effects like hepatotoxicity. Most importantly, it induces immune impairment responses, resulting or we can say that it makes the host more vulnerable to TB reactivation and reinfection. So now we need an alternative therapy that can fight this deadly disease and can reduce the length of treatment or associated side effects and emergence of drug-resistant variants and prevent reactivation of the disease. So we believe that addition of certain immunomodulator can, overcome the shortcomings associated with this ATT. So for this, we screen many immunomodulatory compound for their antimicrobial activity as well as immunomodulatory activity. Here I would like to discuss berberin, which we have just published in which we have just published this year in PLOS Pathogen. So berberin is an alkaloid compound with very weak antibiotic activity. People also have shown its nature. I mean, it can be used as efflux pump inhibitors. It has anti-diabetic nature, and it can also reduces hyperlipidemia. People also have shown its anti-cancerous nature. So we wanted to ask whether berberin has any antimicrobial activity or any immunomodulatory activity. For this, we took THP1 macrophages, monocytes, and we differentiated them into macrophages. And along with, we also took the peritoneal macrophages, murine peritoneal macrophages. And then we infected these macrophages with virulent strain S37RV and treated with berberin along with frontline drug isoniazid. And interestingly, what we observed that berberin treatment significantly reduces the bacterial burden in both the macrophages. And most importantly, we got synergistic effect when we used in combination with isoniazid. So therefore, we can say that berberin potentiates the antimicrobial potential of frontline anti-TB drug isoniazid. So now we got that, yes, in ex vivo setting, this molecule is working perfectly fine. So then we wanted to translate this finding in vivo model, where we infected animals with S37RV. And, after 15 days, we started treating these mice with berberin, isoniazid, and isoniazid plus berberin. And at different time point, we took out the organ to look at the bacterial burden as well as immune responses. So interestingly, what we observed that berberin treatment significantly reduces the bacterial burden in both lung and spleen of not only drug susceptible strain, but also in case of drug resistant strain. So, and most importantly, we got synergistic effect when we used combination therapy of isoniazid and berberin. Therefore, we can say that berberin treatment significantly potentiates the antimicrobial activity of anti-tuberculosis therapy in murine model. Then we looked at the elicited immune response after berberin treatment, and we observed that berberin treatment significantly induces macrophages activation, T cell activation, B cell responses, and significantly heighten the stem cell memory responses, central memory responses, and tissue resident memory responses. And these memory responses are required for the long-term protection against any pathogen. Then we wanted to look at the mechanism of the heightened memory response. As we all know, a naive T cell upon stimulation gives rise to stem cell memory response, which further gives rise to central memory response, effector memory response, and tissue resident memory response. And these memory responses are, required to fight with the pathogen at the tissue specific site. For this, we took human PBMCs from 4 individuals that were PPD positive, and we took human PBMC and treated with CSA, Mycobacterium tuberculosis CSA, that is complete soluble antigen of Mycobacterium tuberculosis, along with berberin. And what we observed that berberin treatment significantly drives the differentiation of naive T cells to effector memory T cells and resident memory T cells. So in order to look at the, why we are getting the heightened immune response, for this, we perform proteomics of the PBMCs derived from these 4 PPD positive individuals. And we observed that berberin treatment significantly alters the proteome landscape of CD4 positive T cells, resulting in the upregulation of diverse signaling pathways, which are required for the protection against TB. So, and interestingly, NOS signaling and glycolysis were upregulated, which is required for the generation and maintenance of tissue resident memory T cells. So, ironically, this NOS signaling activates PTEN molecule, which further inhibits AKT signaling, and which led to the activation of FOXO1 and BLIMP1 that is required for the transcription of TRM specific gene. Then we again further validated with RT-PCR, and we observed that berberine treatment significantly upregulated the TRM specific gene like FOXO1, BLIMP1, NOS3, PTEN, etc. Then we came up with the model that berberine induces glycolysis, which is required for the effector function of T cells. And then we further validated this data by doing bioplex assay, where we observed that almost all the pro-inflammatory cytokines were upregulated after berberine treatment. Now we have proved that yes, berberine is significantly inducing T cell responses, memory responses, not only in mice model but also in human samples. So yes, now it is inducing central memory response. Then therefore we thought, can it increase the vaccine efficacy of BCG if we are treating berberine to BCG immunized animals? For this, we again performed a vaccine experiment wherein BCG vaccinated animals were boosted with berberine, and after a certain period of rest, we infected these animals with virulent strain S37RV through low dose aerosol infection model. And then we at different time point we took out the lung and spleen to look at the bacterial burden and immune responses. And interestingly, what we observed that berberine treatment significantly enhanced the BCG induced antitubercular immunity, as evidenced by reduction in bacterial burden by more than 2 log in lung and spleen of the BCG immunized animals. And also it induces the tissue resident memory cell responses in BCG immunized animals. So we can say that yes, berberine has not only the immunotherapeutic activity, but it has also immunoprophylactic activity. Then, as we discussed that in the previous slide, that ATT induces the immune dampening effect and causes host vulnerability to TB reactivation and reinfection. Then we wanted to look at whether berberine treatment can reduce the TB recurrence. So for this, we perform TB reactivation and reinfection experiment, and we observed that berberine treatment not only reduces the rate of TB reactivation, but it also induces the bacterial burden in the lung and spleen of reinfected animals. So yes, so now we can summarize this finding that of course NOS signaling is known to induce PTEN expression, which in turn activates FOXO1 by inhibiting AKT, and convincingly berberine treatment significantly induces the expression of PTEN and FOXO1 that can induce the immunological memory against TB. So in similar setting, we also performed some experiment to prove that vidaferin A, that is active constituent of Vidania somnifera, that can protect the animal against primary and recurrent TB by modulating mycobacteria specific host productive immunity. This study just got published in Microbiology Spectrum. So on this note, I would like to thank my lab member, my collaborator, ICGB core facility, and of course my funding partner. Without their help, I could not be able to perform this experiment as well as this study. So thank you.
Thank you so much, Dr. Ved. Sharanya, are you there?
I hope I was.
Yes. Yes, it was very nice. Sharanya, are you there?
Yes, sir.
Yeah.
Sir, I had a question. So you particularly emphasized on use of berberine as an adjuvant, but are there any alternatives to that? Like, are there any different adjuvants you're also studying on that also have potential?
We also that table showed only we screened many bioactive compound like it we have like 11 12 molecule that we here we I just showed data from one publication. So just in last slide I showed Vidania somnifera also can be because now in 2020 people started taking Vidania somnifera as prophylactic against COVID. So we thought whether it can protect the it has any potential as anti tuberculosis. So yes, we did the similar experiment what we did for berberine, and yeah, observed the similar result. But yes, we are doing we are formulating many combinations with those 11 12 molecule.
Thank you, sir.
Yeah.
So, Dr. Ved, I have one question. We are looking after the increased efficacy of the BCG vaccine. Have you also tried the same using Mycobacterium indicus prani, which is again a new candidate for vaccine?
That is really good question, but yeah, we can try. We can try because see, what we were thinking that everybody is getting BCG, so we thought first to check with whether it has any potential to increase the efficacy of BCG, but definitely we can try with MIP also.
Sure. And one more. So when you check the reduction of bacterial burden in the lungs of mice, have you also checked whether the bacterial infected mesenchymal stem cells also exist in the bone marrow?
Yeah.
So they exist, right?
The idea was to I mean we can discuss
Yeah.
later, but yeah, idea was to infect the mice with pre-labeled S37RV, and then we can isolate different cells and look at the bacterial burden after treating with these immunomodulators. But yes, the study is very long, and yeah, we are doing, but hopefully I hope we can discuss in next meeting in near future after
Sure.
the
Sure.
publication.
Sure.
So
Thank you.
it's unpublished data. Yeah.
Yeah. Thank you so much.
Mhm.
So, Sharanya, so
Uh,
with this, I would like to thank Dr. Ved Prakash Dwivedi for such a wonderful and insightful talk. I have been knowing him for last 3-4 years, and he's a nice person. Along with that, he's a very good scientist also, and very good immunologist, which are very few in numbers in case of tuberculosis field in India. But there are very few in numbers. Dr. Gobardhan Das is one such great scientist. So I would like to thank the MIT University for the infrastructure, and also the Vice Chancellor sir for giving us the opportunity to organize this event. Dr. Penna Supasanna, who is the Dean of AIB, for supporting us to organize this event, and the organizers, Dr. Abhishek Gulle, Dr. Vidupak Vastikar, and Dr. Ramesh, along with myself, and along with that, Sharanya, for successfully hosting this series for so long. So first talk in this series was given by Professor Sarman Singh. Second talk was given by Dr. Deepak Kaushal. Third talk was given by Dr. Savita Kulkarni in BRC. And the fourth talk now we witness from Dr. Ved Prakash Dwivedi. So thank you so much, Dr. Ved. Thank you once again. Yeah.
Thanks, Shank. Yeah. Thank you.
We'll see you again.
you missed a person yesterday's talk. You missed one
Oh,
talk.
so the yesterday's talk was given by Dr. Anand Krishnan, and today is Dr. Ved Prakash Dwivedi. It's fifth session. So in total, there are 10 sessions in this series.
Thank you so much, Shashank, for giving
Thank
me
you.
this opportunity. Yeah.
Thank you. Yeah.
Okay.
Yeah.
Take
Bye.
care. Bye.